Okay, so it's a pleasure today to have Bjarke Nielsen from Roskilde University and the Niels Bohr Institute. And he's going to talk to us about superspreading, mitigation, and evolution. Please go ahead.
Okay, thanks. And I'd of course like to talk. Thanks, Jay, for inviting me. And he was back in the day, I want to say, but a few years ago, my thesis supervisor, my master's thesis supervisor. So together with Niels Bohr, so it's a pleasure to be here and so on. So yeah, I'm going to talk about superspreading, which is this phenomenon that some diseases have enormously wide distributions of how many infections each person causes, so to speak. But from a sort of slightly statistical physicsy point of view. But before I do that, I'd just like to start by digressing a bit and taking a couple of minutes to talk about a couple of other topics that I think fit in well with the theme of emergent phenomena that also interest me, just in case someone in the audience thinks this is cool and would love to chat to me about it or something. So those two topics are morphogenesis, which is how biological shapes emerge, and the other one, it has to do with chemical patterns, or specifically how polymers interact to create funny larger scale patterns, essentially. So just a quick couple of words about these two other projects that I'm very involved in. So morphogenesis is this sort of funny field of trying to figure out how do biological shapes emerge, usually during embryogenesis, so when the embryo is formed. But you can also study these so-called organoids, which are organ-like structures that can be created in a lab. And the thing about organoids, we see two examples here of stained organoids, is that they sort of on the micro scale or on the smaller scale, they look quite a lot like real organs. But if you sort of zoom out a bit, they don't really have the large scale structure. So while you might get something that looks like gut or something that looks like, brain tissue, for instance, you won't get an entire brain grown in the lab. I mean, that sounds obvious when I say it, but it's important to understand these differences. So, but that's something you can actually do on a controlled scale in the lab. And what I'm interested in doing is sort of trying to model how these things come together from a few very simple rules of purely cell-cell interactions. Nothing about big organizing external forces or anything, but just cell-cell interactions. And I just wanted to show you sort of the super simple model with essentially only a couple of degrees of freedom for each point particle is what it is essentially. You can get something really funny just by starting from completely random conditions. So all this is just that each point particle has, of course, a position, and then it has one like polar vector, one vector quantity associated to it, and that is what is in biology known as the apical basal polarity. So epithelial cells have these polarities, and just by one polarity and starting things from a random initial conditions, you can get some really funny structures. So that's just what I wanted to show you. But we've also used this idea of point cell or point particle modeling of morphogenic processes to model some more sort of specific and realistic processes. One is, for instance, vasculogenesis, which is how the initial vascular plexus in the body forms, and that happens by aggregation. So you start with some number of essentially some dilute cloud of cells, more or less, and then it condenses into a network like tubular structure, and that's something we've also modeled. So that's one of these. I would like to stress sort of the emergence of this thing, that it's very, very simple rules giving rise to some large scale structure that really can perform some job. That's one project. Another project I'm very involved in is about block copolymers. So that's these kind of copolymers that are built up of blocks of two different types of monomers, or in this case, it's just two at least. And they have, differential interactions. So maybe one type of monomer likes being close to another type or the same type. And that can give rise to some really funny large scale or larger scale at least patterns. And of course, they also have some applications in the microelectronics and microfluidics and so on. But my way into this has again been from a modeling perspective, but just trying to see how simple a set of rules can you sort of set up. In this case, it's a continuum description that can really capture how these things crystallize and form patterns on various surfaces. So I just wanted to start by mentioning that if you want to talk about that kind of thing, sort of slightly more soft matter-ish stuff, then I'm also down for that. So moving on to the super spraying story, that's the main thing here today. So it's about how infectious diseases spread, and especially sort of the heterogeneity aspects of that. So not so much the mean field behavior that everybody's talking about, but actually how the heterogeneity comes into the picture. But before I jump too much into it, I just want to, remind you of something that happened, what feels like perhaps long ago now, March 2020, is that we were all presented with this flattening the curve concept. And for some reason in Denmark, it became the red and the green curve. I'm not exactly sure why, but they were all presented, the curves all looked somewhat like this, that either you could have no restrictions and you would have this enormous outbreak, or you could have some restrictions and you would apparently still have a growing epidemic for a long time, as you can tell here, but just never reach the same peak. That was sort of the idea. But what really happened when the lockdowns were instituted was something completely different. And just taking the example of Denmark, that they instituted the lockdown, and now we're looking at confirmed deaths here. And it only took, it was essentially immediate or very fast that you went from a growing epidemic to one that was rapidly declining. So what we would call an R0 value of less than 1. That happened almost immediately. So what happened was in fact very far from what had been predicted. And the models that were used by the authorities at the time could not capture this at all. They tried to put in the altered contact rates due to the lockdowns, and they just could not capture why it would have such an enormous effect. So that sort of led us, when I say us, I mean Kim Sneppen and Lone Simonsen and myself, to think about like, why is this? Why didn't the models capture this essentially? But of course, there are many other interesting types. There are many interesting types of heterogeneities. I'm going to talk about mostly in transmission, so how the disease spreads. But of course, there are also social and contact heterogeneities. Contact network structure is important. Contact network temporality, so the fact that your contact network is not a static thing, but changes over time, also important. Differences in social time and contact rates, that's something else again, right? One contact network has to do with how many friends you have. Social time has to do with how much time you spend with them. That's two different things, right? Then there are biological heterogeneities, differences in how much virus do you shed when you become infected. What does your viral load profile over time look like, for instance? How susceptible are you to the disease? How does the disease progress in terms of symptoms and so on? There's lots of things here. And most of them are not studied that much on a sort of individual or at the scale where individual differences matter, so to speak. And it turns out that individual differences matter a lot in many of these questions. So to take a sort of statistical mechanics viewpoint or statistical physics viewpoint on this, it's very important to distinguish between types of noise or types of disorder. Noise is not just noise. You probably all know this, but it's important to keep in mind that, noise could mean a Nielsen disorder, which is sort of like particles in a gas. You have some sort of variable that's stochastic, but it's fluctuating in time for each entity, so each particle in this case. So you have all the particles going around, and they change velocities once in a while. But overall, you know that the velocity follows some specific distribution. But it's not a fixed property of each particle. So if you measure one particle at one time and the same particle at a later time, it will have a different velocity. The sort of the opposite extreme is Cringe disorder. So that's more like impurities in a material, for instance. So if I look at this crystal and I see that there's a phosphorus atom right here and a boron atom over here, then if I look again tomorrow at the same piece of crystal, the phosphorus atom is still going to be there and not at some other space or some other site in the crystal. So that's more like a Cringe disorder. And what does this have to do with disease modeling? Well, we'll get to that in a moment, but let me just mention that in order to even keep track of any Cringe disorders, you can't really use mean field descriptions. You have to use something like an agent-based or an individual-based model, which allows you to keep track of each individual in the system. And of course, it's true that social interactions, we mentioned that as a kind of heterogeneity before, have a certain monotony to them. So two people are more likely to meet again if they met in the first place and so on. So that means that, again, keeping track of individuals or of repetition is important. But on the other hand, social networks also rewire over time. That's the temporality I talked about before. Also something that can be hard to capture outside an individual-based framework. And then lastly, as I mentioned, there's this inter-individual variation. That's where superspreading comes in. Differences between how individuals spread the disease, for instance. And that could, of course, be an annealed or a cringe disorder sort of ab initio. You don't know a priori what it is. But I'm gonna maybe here describe some reasons to think that it's more quenched than annealed. But that's not hugely important to all that I do here, but it's worth discussing, I think. So the basic fact of COVID-19 is that it's very much a super spreading disease. This is something that has been measured by many different methods by now, from phylogenetic to analysis of aggregated time series and over contact tracing data and so on. And generally, it has been found that something like 10% of infected individuals cause 80% of new infections. And you could say, well, maybe that's just the case for all similar respiratory infectious diseases, but that turns out to not be the case at all. And that's something we can get back to in a second. But to return to the question about, is it then quenched or annealed? So that means, is it something that sticks to the person, or is it more a question of, one moment I'm super infectious, the next moment it's another guy, and then it's some other guy? Well, there are some good arguments for why it's probably somewhat quenched. So the first one, which came really, really early in the pandemic, something we had access to very early, was household transmission data. And that showed that that there was an attack rate within households of just 15%, essentially. That means that if you get the virus, then your chance of transmitting it to some other person in your household is just about 15%. That's not very high. And it sounds like something that couldn't even sustain an epidemic. But when you think about it like this, that some people could become very infectious and infect almost all the household, and then a lot of people, to keep the same average, what we call the R0 value, there must also be a lot of people who don't become infectious at all, essentially. And that can explain the 15%. But if you think about it as a annealed thing, that it's just that your infectiousness varies a lot throughout the disease, you can't really explain the household transmission pattern, right? Because you're pretty much bound to come into contact with the people you live with, so that the annealed description cannot really explain that. And the other fact is that overdispersion, so that's this super spreading degree, the tendency towards super spreading, varies enormously between diseases that are otherwise quite comparable. So that also means that a behavioral explanation is quite unlikely, because people are not that likely to behave drastically different whether they get pandemic influenza, for instance, or whether they get COVID. So that also seems slightly unlikely that that's at least the main or the driver, since there are so big differences between comparable diseases. And lastly, the viral load variability, so that how much virus do you actually get in your body, the variability in that is very correlated with the super spreading tendency of different diseases. So some diseases have very high viral load variability, like COVID or SARS-CoV-2, and others don't. But actually, this goes back to even the late 50s or early 60s studies on this. And the first one that I know about, or at least very classic experiment, was actually on tuberculosis, and it's this brilliant experiment where they had essentially different rooms of tuberculosis patients set up, and then they took the air from those rooms and let them into chambers with guinea pigs, and then they saw how many guinea pigs caught tuberculosis. That's a classic one. It's actually quite ingenious if you think about it. But there they found that just 3 out of 77 patients, even though they were all smear positive for tuberculosis, but just 3 out of 77 patients caused 73% of infections among these guinea pigs in the end. So that's also a kind of super spreading. a very wide distribution of subsequent infections.
Sorry,
But,
Bjarke, can I
yep.
just ask,
Sure.
so what you're saying is that COVID transmission rates are likely just to be because of a few people who are super spreaders? Is that?
Yeah, essentially that it's super variable, at least that many people will not infect anyone, even if they catch COVID, and even if they are around other people. But some people become enormously infectious and can infect many people at one event. That's what it looks like.
And then you also claim that this has nothing to do with like social context, like the amount of social context, or the amount.
Well, no, I'm not claiming it has not. No. I wouldn't claim that it has nothing to do with that. That's, I mean, it's more like it's a means and opportunity kind of thing, right? The means is the high infectiousness, and the opportunity is, of course, contact. So of course you need to have means and opportunity to be an effective super spreader, so to speak.
Yeah, of course.
And who are these people?
Yeah, well, that's actually not well known. There is a study that shows that there is quite some that there's some correlations with age and with body mass and other things, but it's not like you can single out, who it is. But people have looked into it a bit. But the sort of the most salient fact here, I think, is that some diseases just have this enormous variation in how much virus is created, and we don't know exactly the mechanism, I think.
So I've one more question. Can you hear me?
Yeah, sure.
Yeah. My understanding is that when you do PCR tests, that actually there is the threshold of detection depends on the amount of amplification cycles. And that's a parameter that's generally not disclosed to people who get tested. But as a researcher, do you have access to those distributions? Because that would give you a sense of the percent of the distribution of viral load during testing. And
correct, yes.
And are those the spreaders who trigger like 15 amplification or something?
I'll show you a couple of distributions in a second, actually, but that's completely correct. Maybe I should explain a bit more about how the PCR works, but that's essentially, it's the case that whatever, you have some amount of virus present in the sample originally, and then the so-called CT value is how many times do you have to double this amount before it is actually detectable. So if you have to double many times, that means there was very little originally. And if you have to double only a few times, that means it was a large amount. That's what this so-called CT value measures.
But is that also the super? Do we know that those are the super spreaders, or they just have high viral load? That super spreading is not the same as high viral load, or is it the same thing?
The best understanding is that it's almost the same thing, at least.
Okay,
it's very highly correlated.
Super.
But yeah. So. But yeah, this super spreading, or at least super spreader phenomenon, was somewhat controversial, or is maybe still somewhat controversial. I mean, we struggled a bit with the epidemiological community, but I think the consensus seems to be shifting nowadays. And there's this recent paper in Lancet Infectious Diseases where I think they explain it quite well, where they compare the degree of super spreading in influenza disease and in COVID-19. And they say that inherently most COVID-19 cases are minimally infectious, so most of them don't really infect. But highly infectious individuals are estimated to expel hundreds to thousands of virions, so that's virus particles, per minute while talking, singing, or coughing. Meanwhile, a greater proportion of individuals infected with H1N1, so that's an influenza strain, are inherently infectious but expel virions at low rates. So in general, the influenza, it's more democratically distributed. The rates are somewhat lower, even though the average infectiousness may be the same, right? And in COVID-19, it's sort of the case that most people become quite uninfectious, really. So that I think is well explained. And then there's another recent paper in PNAS that just the main message is essentially this: that just 2% of SARS-CoV-2 positive individuals carry 90% of the virus circulating in communities. That's one of the most extreme findings I've seen. There are some others we'll look at in a second, but that one is quite extreme. And then we'll just look at this figure that I cooked together, but based on some data from a study by Kidd et al. That sort of shows that here you have the orange bars is just the viral load histogram on a log scale here, log x-axis. But what's really sort of maybe even more salient is this, the red curve, which is the cumulative, simply the cumulative amount of or number of virions, or percentage of the total virions. So what you can see here is that 10%, you can actually not see that, but I'm telling you that 10% of positive individuals end up harboring 90% of virions with this distribution here. And that's a measured distribution of viral loads. That's also extremely wide. Of course, it doesn't look that wide because it's on a log scale, right? So it looks like it isn't that wide, but it's amazingly wide when you think about it. So in another way of putting that is maybe more physicsy, is that imagine you had taken all the viral particles and pooled them together. You pick a random one, then you're actually 90% sure that you'll get one from this most infectious 10% of people. That's another way of putting it. So in order to model something like this, we wrote down essentially an agent-based model. I'll get into that a little bit more. But we model the super spreading aspect itself, or the biological infectiousness, as something you draw from a gamma distribution. So that has some average value, and then it has this dispersion factor called K. And the lower the dispersion factor, the wider the distribution. So high K is something like this. You see the K equals 10 here. That doesn't look very extreme, but at low K values, such as 0.1, for instance, you get something that diverges near the origin and then has a very long tail. And that's the 0.1 is quite close to the estimates generally seen for COVID-19. So that's a very broad distribution indeed. And that's, when I say that, I mean the 0.1, that's for the actual distribution of the number of infections that each person causes, not the viral load, just to put that out there. But then on top of this, so each individual has some sort of infectiousness drawn from this distribution once they become infected. And on top of this, it's sort of a quite simple model. You have some networked contact structure, and the precise network is something we can discuss. And then social time is assumed to be a finite resource. That turns out to be quite important, because previous models have often assumed that super spreading is essentially a question of some people having enormous social networks, some people having 100 times the average or something social contacts, different social contacts. But then of course you run into the question of whether it's realistic that you can actually also spend a meaningful amount of time with each of them. So in this case, we make sure that social time is some sort of finite resource. And then the disease progression itself is just what is quite well known from the so-called SIR or SEIR models, that you start out as susceptible and then you may become exposed by some other infectious individual with some rate when you are in close contact or in contact. And then you progress through the stages. So there's an exposed stage, and it's split into 3 for reasons that are not important here. And then you progress to the infect stage, and then in the end you either recover or die. So that's what it is. And we don't distinguish between recovering or dying, because we're not actually that interested in outcomes here. It's just the spreading that's interesting.
What is the role of the pre-symptomatic in the model? Yeah,
there's also a stage that's called pre-symptomatic, but it's irrelevant for what I'm going to discuss now. But it's important if you are trying to model things like contact tracing, because then you're really interested in whether people actually, are aware that they have the disease or not. So then you need to include the pre-symptomatic infection stage. But here you can just imagine that it's exactly the same as the infected stage. Okay. So it doesn't matter for this actually. Okay. It's just nice if you want to discuss some contact tracing aspects. Okay. But for now, it doesn't mean anything. So what happens if you just you take a model like this, and even just in a well-mixed population? So imagine that there's no contact network. It's just everybody meets everybody. What kind of infection networks or infection chains do you then end up seeing? You can almost imagine, I guess, that if you have a very super spready disease, so like this one on the right, that's an infection network from a disease parameterized to look like COVID-19. Then you get this this network that sort of has some enormous hubs, and also that you can see that most secondary cases, so these yellow ones, they actually don't end up infecting anyone. It's only that once in a while you see a big outbreak. That's of course fairly obvious. And in the opposite extreme, taking this dispersion parameter k to infinity, so that means completely deterministic spreading, or at least Poissonian statistics in this case, then you get a sort of a tree structure that is much more democratic, that most people have a few offspring. But of course, both of these are finite outbreaks that, you know, they don't involve more people than this, so they have to end somewhere, of course. So at the end of the branches, you will see that nobody infects anyone, of course.
There's like a patient zero in these networks?
Yes, there is a patient zero somewhere, but I didn't mark him, so I cannot actually tell you who it is.
It just looks like in the one on the right is the actual middle.
Yeah, it could be. Yeah, could be.
But normally you model like this, or you model several patient zeros getting it simultaneously. And
Normally, I will model several. I just picked out of a simulation. I just took one infection chain and studied it just for illustration. Yeah.
So just to make sure I understand, so the main difference with the more traditional models is that the infection rate is itself a random variable that you every time.
Yes.
Like
Exactly. Yes.
Has a different one.
Yes.
Or
Exactly.
every time
Yes.
somebody gets infected has a different one.
Yes, and that and when you then draw the infectiousness, then that actually stays with you for the duration of the disease. So that's why you need to keep track of people, essentially. Well, that's one reason.
So the infection rate is fixed for each node?
Each person, yeah, exactly.
Yeah. Okay.
So that's the quenched part of it, as opposed to annealed.
You're not varying it in time. Yeah.
Exactly, yeah. And you're not, and because you could make a model where everybody varies enormously in time, so you still get this over dispersion, but it's just because that each person, you know, on day one is super infectious and then on day two is not, and so on. You could do that. That would be an annealed kind of situation. That would give some different results.
Yeah, but like you were trying to argue, is not the right thing.
No, exactly, yeah.
And so from the right hand side in this picture, C, that would be like the classic homogeneous SIR type models.
Exactly,
and
yes.
The left hand side would be when you pick from this distributions that's heavily skewed towards.
Yeah, exactly, yes. And this is even just in a well-mixed population. So when everybody just, you know, pops around in some soup, meeting all the other soup particles, like a well-mixed situation.
But then it's not about the structure of the network. It's just that the nodes are different.
Exactly. So in this case, it's not about the structure of the network at all. I'll add some network structure in a moment, but here it's actually there's no network essentially, not yet. There's only this. This is just the infection network. So that's just something that emerges from running.
So to mimic this super spreading, you actually is the network structure not really important? It's just more like each node should have different varying infectious rates.
Yeah, that's the main thing. That's the super spreading part. But actually, it turns out that the interaction between this super spreading, so the variable number of, or the variable infectivity, and the network structure is enormously strong. So we'll see that in a moment, that if you add network structure on top of this, then you get something that's very, very interesting.
Okay.
So, we'll see that in a moment. But I just wanted to make one oh, sorry. Was there another question?
Sorry. So I mean, I heard you said that it's well mixed, but if it's well mixed, you cannot really keep track of the individuals, right? So then I'm a bit confused what the nodes. Really
Oh, well mixed. Well mixed just means that everybody has the same chance of meeting everybody else.
Okay.
But I can still keep track of them in the computer, right? That the person I is highly infectious and person I+1 is less infectious, and so on. I can keep track of that still. The well mixed just means that everybody has the same chance of meeting everybody else, essentially.
Yeah, you're basically saying there's no network there.
Yeah, there's no network. Let's just imagine every time step you take two people and then you say these guys met. I don't care if they know each other. I just assert that they met for some reason. And then in the next time step, it's two other people, for instance. That's the simplest way of imagining it, I think. So, but I just wanted to comment on the middle part here, because that's maybe even more interesting, which is sort of the dispersion parameter for for a few different respiratory pandemic threats, so to speak. And that's on the x-axis, and on the y-axis you have this metric of how large a fraction does it take to account for 80% of infections. So that's another way of measuring this super spreading tendency, but they are very closely related. Actually, it turns out that this k, the dispersion parameter of the gamma distribution, and the fraction responsible for 80% are almost, they are essentially equal up to about 25% or something. So that means that if you have a K value of 0.1 like COVID, that means 10% account for 80% of infections and so on. But for the flu, on the other hand, K is more like 1. And that means you're quite close to homogeneous actually. Of course, you will never get that 80% account for 80% of infections, because even in the case where infectiousness, so that's the means and opportunity I talked about before, even if the means are completely evenly distributed, the opportunity will never exactly be. And that, to be sort of mathematical about it, that means that if you draw infectiousness from a gamma distribution, then to get the actual number of subsequent infections, you need to then draw that from a Poisson distribution whose average is determined by the gamma distribution. And that's mathematically the same as drawing from a negative binomial. And in the limit of infinite K, the negative binomial becomes the Poisson distribution. So that means that even in completely homogeneous case, you still have a Poisson distribution. So there's still some variance. So you never get 80% causing 80% of infections, even with the most homogeneous disease you can imagine, essentially. So, so yes, this is sort of this. You can see our PRL paper for more details on this if you want. But actually, we can sort of take a bit of a step back in terms of complexity and drop all the agent-based, call it, and just think about this analytically for a moment, just statistically. So let's imagine that we make a simple analytical approximation. So now you have some number of neighbors that you draw from a distribution. And that could of course be infinite, but let's just say you draw a number C from a distribution, and you also draw your own infectiousness or your biological infectiousness that I call little r from some other distribution, pi. And I imagine that that's a gamma distribution. Then if
sorry, what is mu and k there?
So mu is just the average infectiousness, whatever it is.
Okay.
And k is this dispersion parameter of the gamma distribution. So that measures how wide is the distribution. Low k means very wide, and high k means deterministic, essentially. So actually, in the limit of infinite k, the gamma distribution becomes the Dirac delta. So that makes sense. Okay, so if you know the C, so the number of neighbors that you have, or the degree if you're talking sort of in a network lingo, and you know your infectivity, then you can compute the expected, or rather the distribution of your reproductive number, so the number of persons that you will infect, that I call capital R, according to this formula, which is pretty easy to derive. But then on the other hand, if C, so the number of neighbors, and R are distributed according to these distributions I mentioned, then you can integrate them out, and the expected, or rather distribution of offspring or of reproductive number is this integral. And then if you really simplify things and say that the degree distribution is just a Kronecker delta, so that everybody has the same number of neighbors, then you get this funny sort of equation for the average reproductive number that you actually see in the population. And the equation looks sort of funny, but we can just go ahead and plot it, what this means. So let's just take it in a few steps. So here I have the histograms in the top panel here. It's just for a homogeneous disease. So that's just taking k to infinity. Then what you find is that if you just let the structure be well mixed, so that's having an infinite number of contacts, then you get an R naught. So this average number of actual infections happening here of 3. That's just how I parameterized it to look a bit like COVID classic, so to speak. And then if you reduce the number of contacts to 10 and then down to 4, you get very moderate reductions in R naught. Even if you go down to just having only 4 contacts ever, you can still have an R naught of 1.4. So this is what I showed you with the green and the red curve, right? And the classic SIR models, that I mean, you can't really, in the k equals infinity case, you cannot explain why the contact reduction really has such a large impact on the epidemic, because you can see the histograms look very similar. But once you go to the super spreading case, that's the bottom panel, then for the well-mixed case, you have this enormously wide distribution of offspring or of reproductive numbers with an average still of 3. And then when you start reducing the number of contacts down to 10 and then down to 4, you really reduce, first of all, you change the histograms enormously. And also, as you reduce it down to just 10, you have essentially reached the critical point, almost at least, of R = 1, whereas in the homogeneous case, it was still at 2.3. So this reduction in contacts now has a much larger influence when you are in the super spreading regime than it had in the homogeneous spreading regime. And over here, it's just a contour plot showing essentially the same thing, but just more systematically. But this was all just based on what I showed you on the previous slide. No agent-based fanciness here, but you could run the agent-based model, and I tested that, and it gives the same results, or very, very nearly in these cases. So now, but what we saw before can of course not tell us anything about the time course. So let's just move on to.
Sorry, can I just ask, because I got a bit lost here. So what is the difference between the bottom plot and the top plot? I mean, in terms of the equations,
so the bottom plot is the one where you have super spreading. So that's k equal to 0.1, like for COVID. And the top plot is deterministic essentially, or that's k equal to infinity. So like more like a SEER model essentially.
So the k is measuring how the distribution of the rate of infection, right?
Yeah, exactly. Yeah, so high k means homogeneous, or
like everyone has the same rate.
Everybody has the same.
Okay, all right,
yeah.
all right.
And in the bottom, it's very, very different between individuals, but at a COVID-like level essentially.
But then you look at the top plot and you say, well, this is not really, it's not really it. But why do you say this is not really it?
Because when you start reducing the contacts, so that's what happens from the purple to the red to the orange. Yeah. You only get very moderate reductions in the R0, the actual average number of persons that each individual infects, and that's exactly like what we saw in the very beginning here. That they assumed that once you institute a lockdown, you just get this still increasing epidemic, but just at a slower pace. But that's not what we saw in real life. What we saw in real life was that you instituted the lockdown, and it had a very large effect, very, very quickly.
I see.
So that's and we know now that COVID is a super spreading disease, and we can see that if you incorporate super spreading, then reducing contacts all of a sudden has an enormous effect. You can just decimate this R0.
I see.
So somehow now the sensitivity to contact reduction is much greater when you have super spreading. And if you want to look at the time course of what this looks like, then just it's the same as, I mean, the idea is the same as the previous plot, but now just the time course. Then in the case without super spreading, so that's here on the left, you get smaller epidemics, but not by that much, by these moderate contact reductions. But in the super spreading case, you can bring it right down by the same reductions. So that's just showing the same at a time in a time evolution. So, but what about network structure as we talked about before? So first of all, the size of the network, that's essentially what we had here. That was enormously important, we could see. So what's the average degree, I should say, in the network is really important. But what about the network structure? So in the top plot here, I assume an Erdős-Renyi network. So that's like the least networky network that you can have essentially. So I have an example of a little Erdős-Renyi network out here, where what happens in an Erdős-Renyi network is that every person has a Poisson distributed number of neighbors. So it's relatively narrow distribution. Most people have around the average. And you don't have any sort of triangles forming. That means you don't have any clusters. So in an Erdős-Renyi network, it's never the case that the friends of your friends are also your friends. That doesn't really happen in an Erdős-Renyi network. And actually the probability of it happening falls off as the size of the network grows larger. So in the infinite size case, it never ever happens. So I'll just get to the plots in a moment, but let me just explain the bottom network instead. So here we have a network with a very high degree of clustering, and way that it's generated is that you essentially assert that each person is a member of two groups, and each group is completely internally connected. And we can discuss sort of the exact algorithm, but that's the idea. So that gives you just that every person essentially knows two groups of people who all know each other internally inside each group. That gives you a very high degree of clustering. And what we show on the contour plots is then from the agent-based simulations that you have the K, this dispersion factor, a parameter on one axis, and you have the attack rate in the color scheme, and then on the other axis you have the number of contacts. So it's a bit of a dense plot. But what the idea is that as the dispersion parameter becomes low, so it begins to look like COVID, which is at 0.1 essentially, then you see that you can move to a much higher number of contacts before the attack rate starts increasing above essentially 0. So that just shows you again that super spreading diseases are much more sensitive to contact numbers. But in the clustered case, you can just see, first of all, you can see across the board that clustering really reduces the attack rate, but also that it seems to move this, the threshold, the number of contacts you can have before the epidemic picks up, it moves it to way higher numbers, and especially so for the super spreading case, where you can see that at 0.1, you don't even really reach that point in this before you reach the edge of the diagram. So yeah, that means that clustering is also really important. So the actual structure of the network is that's sort of enhanced by super spreading, the sensitivity to that. And then I think I'll go quickly over this, but I just wanted to say that we also sort of expanded this approach into something more realistic, where we took age structure from statistics, Denmark in this case, and also socially sort of age structured social activity patterns from another study by Mosong et al. And then segregated society into, or not society as such, but each person had close contacts and some sort of, which they met regularly or even very often, and some regular contacts such as workplace and school contacts that they met somewhat, also very often, but they were bigger circles of contact. And then they had random contacts, which are just like essentially a well-mixed component. So that's people you meet in public transport or in the public space and so on. And then from the Mosong paper, we could essentially see that people actually spend more or less the same amount of time sort of integrated up over, how many people it involves. That same amount of social time in these three spheres. That's what came out of that study essentially. So of course you can maybe meet many more people in the random sector, but you spend less time with each of them and so on. So we did this, and that gave rise to this. This is just a little chunk of it, complicated kind of network structure where you have young people are these bright nodes and older people are the darker nodes, and they have different kinds of connections, whether they are close, regular, or the random ones are not shown here, but you have regular contacts for adults and for children. And for children, you of course have entire school classes and all that hullabaloo. But I don't want to get into the details too much, because I think, this being the Dutch Institute of Emerging Phenomena, we're more interested in the concept here, I think. But anyway, we also did this.
Sorry, Bjarke, what is the isolated chains then?
So these, so you have, for instance, let's say these two guys over here. So they are, you can see by the color of the nodes that they are very old. So they are, so they are not, they have some close connections.
Not that much, okay.
So they don't get there. So they are probably some old married couple, and they don't have a workplace associated or anything, but they will still have random contacts, which I don't show here because they are not really in a network structure. So they will still have those. So it's not like the epidemic can never reach them or anything. That would be a problem. But they don't have a school to go to, for instance. Yeah. But of course, this is just a section. Yeah.
Random contacts are just homogeneous.
Yeah, they are just assumed to be well mixed. Exactly. So that's the approximation here. And you can always discuss, you can always go more fine-grained, and that's the thing with these kind of things, right? Someone will always come along and say, yes, but what about hairdressers? Where do they fit in, or something? And that's, of course, what level of detail.
I guess it doesn't matter so much, I imagine.
Yeah, exactly. If the details matter that much, then we're on shaky ground, I think.
Yeah.
So Exactly. So, but the concept here, it's not so different, but what we just find here is that the random contacts are really the ones that matter in a super spreading disease. So if you start with a non-super spreading disease, you can try and restrict either the regular contacts or the random contacts. I mean, you can't really restrict the household contacts, right? So we don't bother with that. But what you see without super spreading is that whether you restrict one or the other type has sort of a moderate impact. That's what you see. But in a super spreading case, you can see that restricting the regular contacts has, again, a very moderate impact, like even less than before actually. But restricting the random contacts, that's where you really get the bonus, so to speak. That's where it really pays off.
Close by restaurants.
Yeah, exactly. That's where that idea essentially comes from. It's the restaurant and it's the so on. So
okay, so events, restaurants, etc.
yeah, exactly.
Just work and stay at home.
It's my fault, all of it. No, but anyway. So yeah, that's what you see here, and it makes sense, right? Because I think I'll just get to this. I tried putting it, or at least actually Kim Sneppen tried putting it succinctly in a sentence, which is that it's all about, the number of contacts versus how much contact time you have. That's two different things. So in a super spreading disease, it's the diversity of contacts, so the number of different people that you meet, that matters. And in a non-super spreading disease, it's the contact time. So there, that explains a lot when you think about it, that it's sort of, in a super spreading disease, some people are so highly infectious that they can infect almost everybody they meet, even for a short time. So that's the number of people that you meet that matters. But in a non-super spreading disease, everybody is just moderately infectious, or everybody who becomes infected. So that means that you can infect someone, but only after spending a bit of time with them, essentially. So yeah, that's the thing, sort of diversity of contacts versus contact time. In each type of disease, it's a different thing that matters. And I think if you think about that for a little while, then that explains all the results in one little neat box. Yeah, so maybe I should ask if there are any questions to this part, because I'm going on to something that's a bit more evolutionary in a second, but also super spreading.
No, I just wanted to ask, now joking aside, whether this sort of analysis and stuff had any effect on policy, or this was a posteriori.
Well, that's a good question. I mean, Kim Sneppen and Lone Simonsen, who I did these two studies with, were part of this this body of people advising the government on all of these things. But I think that they were also feeling some degree of frustration that maybe actually didn't have that much of a bearing, or as much as they felt that it should have had, because that was mainly dominated by these SIR type homogeneous mixing models that we all know. So I'm not sure exactly to what extent it had, but at least I know that it had some impact last winter when they were discussing whether to, whether people should go to church in large numbers and on Christmas and so on. There it at least had some impact, I know, because I was a bit involved in that and so on. But the extent of it, I can't really say, but at least some. Yeah. But it's hard for me to tell how much. So I'm just moving on to something sort of slightly newer that I'm thinking about at the moment, and that I
may
think
I ask another question?
Yeah, sorry. Yep.
Okay. First, thank you for your talking. It's very interesting. I have a question. Is about, can you go back one slide, one page?
Oh yeah, sure.
One page back. Yeah. How?
This one?
No.
Okay.
Later. How do you later?
Oh.
Next one. Yeah. How do you modeling the context time when you do agent-based modeling? I mean, I guess if you apply SIR model, then you can increase the for longer context time for the agent, then the probability from jump from the state S to I will be higher.
Yeah.
That's exactly. Just a test, but that's how you do.
That is exactly true in a SIR model, but the problem with the SIR model is that contact time and the number of different contacts, they are put into the same number, right? They are both part of this beta parameter, or whatever you want to call it. But here we can do something else, right? Because here we can say, let's say that I have 5 different friends, And
Yes.
then, so one question is that I have 5 friends, that's one number, but the other is how often do I actually meet them? So that just means that in every time step, I have some sort of probability of making contact with each of my friends. And that probability you could tune up or down. So here there's 2 numbers instead of just one.
I see.
Number of different
So,
people versus number of time spent with each person. Yeah.
so you reflected the contact time as the probability that I contact to my friends.
Yes, per unit time or something. Yeah, exactly.
Okay, nice.
Yeah,
That's
cool.
it. Thank
Yeah.
you.
No, yeah, cool. Thanks. So just moving on, at least slightly, we sort of started thinking about super spreading in a wider context and sort of how does it affect the fitness of a disease. So let's now move on to viewing it a little bit from the point of view of the pathogen instead. Is it a good idea or bad idea for a virus to be a super spreader, in the sense that you have this wide distribution? Does it give a competitive advantage? And I'll get to the motivation of thinking about that in a second. But first of all, when I talk about fitness and competitive advantage, it's a bit of a vague term. It could mean more than one thing. And one thing is that it could mean the ability to avoid stochastic extinction. So that has to do with, imagine you're just a pathogen jumping from a pangolin or whatever to a human. Then what's your chance of actually making it in this world, so to speak? And what's your chance of just that the infection chain just dies out after a few steps, or maybe even before you make it to another human being? That's stochastic extinction. Some viruses could be better at avoiding that than others. And the other one has to do with the ability to reproduce effectively in a population once you have really made it past that initial stochastic extinction risk. So when some critical number of people in the population have already been infected, at what rate does the infection then continue, essentially? And you could say, is super spreading perhaps selected for, or is it selected against evolutionarily? And do mitigation strategies, so the things that we do they change the fitness landscape? Do they change whether it's advantageous or not? It's maybe slightly, maybe it sounds like a slightly obvious step, or maybe not, I don't know, based on what we just discussed. But there was actually a very concrete reason we started thinking about it, which is that a paper came out that just essentially what they did was they were trying to measure how much more infectious the alpha variant, so the one that we used to call the British variant, how much more infectious was it than the ancestral strain, as you called it. And they were mostly interested in the median or the mean even, so the difference in viral loads just on average between people with the new and the old variant. But if we actually look at the distributions in themselves and not just the mean, so let me just explain what we're looking at here, then we can see some more details. So what we're looking at here is how it was represented in that paper. So you have the CT value on the y-axis. So the CT value is this thing, it's this question of how many replication cycles do you have to run in a PCR test before you see a signal. So that means that the higher the CT value, the lower the viral load, but even that, but actually exponentially so, right? Because you're doubling every time. So so that means that a CT value of 10 will have will represent a viral load that's 1,000 times as big as a CT value of 20, because 2 to the 10th is of course around 1,000, 1,024 or something. So that means that, you know, jumping from 10 to 35 is 10 to 30, for instance, is a factor of 1,000,000, right? That's an enormous difference in viral load. But what we are then seeing here is the so-called S negative and S positive, and that S negative essentially means that it's the new variant, this British variant that was new at the time, and S positive means that it's the old variant. And I can get into the details of why that is, but it has something to do with whether you actually detect the presence of the original spike protein or not. That's why it's called S positive and S negative. But what we generally see is that, as you can see, this bar here says that the mean viral load, or the mean CT value actually in this case, has increased. So the mean CT value has decreased, the mean viral load has then increased, of course. It's a bit annoying, this way of parameterizing things. But that we know, that variant was more infectious. We've all heard about that. But what's less obvious from this, and extremely less obvious because it's this logarithmic scale, is that actually the variance, the relative spread in CT values has actually decreased by quite a lot. And that's not clear from there, but if you crunch the numbers, that's what you find, that actually the relative spread, so what's known as the coefficient of variation, is actually about half with the British variant compared to the previous one. So that would seem to suggest that it would be less super spready than it was before, that now the variance in viral load has decreased. That's at least one interpretation. So let's just try and think about what that would mean if different variants had different super spreading characteristics. So what I try and look at here is the first of the concept I talk about, so the stochastic extinction one. So that's sort of how would super spreading affect your ability to make it as a new virus moving into the human population. And I put some dots. So I put the ancestral variant down here. So that's this K value of 0.1 and the mean infectiousness and R0 of about 3. So that's where that is. And if you just focus on the top plot for a moment, then the color just shows the survival chance. So that means that if one patient or one person gets infected, let's say by a pangolin or whatever, what's the chance that the disease will propagate? And what I mean by propagate is sort of, in principle, it just means that it doesn't die out in infinite time in an infinite population. That's sort of the physics way of thinking about it. We can get into that in a moment. But you can also just think about it that it survives for some critical number of generations. If it has survived for 10 generations, it's not likely to go stochastically extinct anyway. So what do we see? We see that low dispersion here, or rather low dispersion parameter, so that means high super spreadingness, is really bad for the survival chance. The survival chance, this is the 5% line here. And what you can really see is that this ancestral variant is actually low in survival chance, something like 15% or something. So that means that 85% of infection chains will just die out. But if you had a much more homogeneous disease, even with the same mean infectiousness, then it would have a much higher chance of surviving. So that seems like super spreading is a very bad idea, seen from that perspective. And in the bottom here, I'm just doing the same thing, but I just try to see what happens if I reduce the activity of each person so that it's not well mixed anymore. You don't have an infinite number of friends, but you just have some finite number, 10 for instance. Then what you just see is that it becomes even more true that super spreading is a terrible idea. And that makes sense, right? Because it's like super spreading gives you this wide distribution of infections. And then you're saying, now I'm truncating it at 10. So that, of course, you're losing the tail. So it makes sense. But so from this, it seems like super spreading would be a terrible idea.
But how do you get these? I don't understand exactly how you get these conclusions. Like, how do you model the extinction? Like,
I can give you a really nice answer actually, because it's so simple. One that's also nicely mathematical. So you can actually, this extinction process, or rather the spreading process, is just this. It's a Galton-Watson process. So it's just a branching process essentially. One thing gives rise to two new things that gives rise to two new things and so on. But except that they don't always give rise to two new things, but there is some distribution, right? So if you have an offspring distribution called Pn, so that your probability of infecting n people, then you can just say, okay, if I want to compute the probability of one infection chain starting with one person dying out in infinite time, then I just say that I call that D, and there's a number of ways it can die out, right? Either it can die out because I infect 0 people, then it's immediately dead. So that's P0. Or it can die out because I infect 1 person, and then that chain dies out. Or because I infect 2 people, and then those 2 chains independently die out. So that's the P2 D squared term. So as in the end, right, you just get the sum over Pn times D to the nth, which is just the generating function of the offspring distribution Pn. So really the equation you're solving is just that D equals, let's call it G of D, the generating function evaluated at D. You can solve that for D. That's the extinction risk. And that actually gives you these plots.
Yeah, I
I've
guess my
also
question
done it in a
is-
agent-based
it's-
model, but it gives the same.
Yeah, I guess I'm just asking what is the parameters you're putting in to model this?
Oh yeah, so that's PN, this offspring distribution that contains all the information.
I see. So you give that, and then you find this stuff. Is that what you mean?
Yep. Yeah.
Yeah.
So it's super duper simple in really, it's super simple. And then you could say, how did I get to the connectivity equal 10 stuff? Well, I can either run it on a computer, but I can actually also just say I suitably modify this PN so that you actually have no more than 10 people to infect. And that just goes back to choosing this the like PN to be something like this distribution. Okay, there's no N in here, but you get what I'm saying, that we had this distribution of how many do you infect, given that you have a number of neighbors and so on. So I can just put that in, and then I can create the bottom plot here using this nice equation here. So that's really neat, really sort of super simple and neat, and not agent based and annoying. So to
Yeah, but
speak.
then the question is why is the virus becoming a super spreader if it's
Yeah, exactly. Why is it a super spreader? That's very strange. But apparently the new variant was becoming less of a super spreader, though. It seems that it's not that new anymore, but the alpha variant, this B117 or British variant, was less super spreading than the previous one before that. And it also, the preliminary data also seemed to suggest that the Delta variant was also less super spreading than the previous
so
one.
is this because it realized it of course wants to be a super spreader to spread as much as possible, but then comes lockdowns, extinguish it, and he sort of has to have a different strategy to get more homogeneous spreading somehow.
Yeah, exactly. That's at least the idea that actually that's one of our takeaways here, is that the lockdowns actually pressure the virus to develop in some specific direction.
Right.
So that, yeah. So what we do really has
Got to become
a
more
funny
homogeneous,
impact. Yeah.
basically, right?
Yeah, more homogeneous. Yeah, exactly.
Yeah.
Because that's the best you can be during a lockdown, at least. It's better to be homogeneous. So yeah. And that's actually, now you just took my conclusion, but that's essentially where we're going. But I'll just show the same thing in a million ways now, in just a few ways. So let's just take Jay's comment, but let's just imagine first that you have two strains now. So you have one that's the dominating, that's the red one here in this plot, the dominating at the outset at least, and then you have a newcomer, that's this purple one. But the newcomer variant is less of a super spreader. So that means the K is a bit higher, but less of a super spreader than the original one. Then in the beginning here, I just assume that we're under lockdown. And then at some point, I remove the lockdown. I'll get into the details of that in a moment. But what you can see here, it's not super clear because you sort of need a logarithmic scale to follow this newcomer variant. So let's go to the next plot where it's the same, but we're just on the logarithmic scale. Then you can see that the old variant under this lockdown, so that's the super spreading variant, is having a hard time and is more or less constant, but the new variant grows exponentially, of course linearly on this plot, but it's a logarithmic axis, right? So it's growing exponentially all the time, that the other one is just constant, even though I have assumed the same mean infectiousness. So it's not like it has an average in the mean, so to speak. And that we can see when I open up society. So when I say open up society, I just mean, you go from having just 10 or 15 contacts, I forget right now, to well mixed. So that means an infinite number of contacts. And what you can then see is that the two variants, they spread equally well, right? You see the slopes just become the same all of a sudden. So then it loses, the homogeneous variant sort of loses its edge again. Suddenly it's not a particular advantage to be homogeneous. And you can, in the lower plot, that's just another way of putting the same thing. That is that if you look at the fraction of the cases that are due to each variant, then in the beginning, the new variant is rising. But then when you open up society, so to speak, then it stabilizes. So they both stabilize. So there's no evolutionary advantage to being either one or the other, super spreading or non-super spreading. So that gives you again this idea that it was really, it depends on what restrictions we impose, what's evolutionary favored.
But what is the best then? So is it the best? Is that an answer to that question? Like, is it the best to do like these crazy lockdowns, extinguish the super spreading thing, and then, or is it best not to do lockdowns and then he'll just spread homogeneous, and or you need to force it to become homogeneous? I guess, yeah, I don't know.
I guess it's a good question, but I'm not sure, because of course this is like a one factor analysis, right? Of course, if we don't have the lockdowns, then and a lot of other things will happen, and of course super spreading is not the only thing that can evolve. So yeah, so I don't know that you can make a conclusion about that, but in this case, I mean, all that it shows is just that if you try and use lockdowns, which are really effective against the super spreading disease, then it has a lot of pressure to evolve out of it, I guess. But I know you already get that.
Yeah,
but whether it's better, whether one is better than the other, I'm not sure.
Okay.
Because once you open up society, it actually doesn't make that much of a difference.
Right. Is that what you showed? It stabilizes both.
Yeah, exactly. So
yeah, but I guess the end product, what is the end? Is it, yeah, but yeah, I guess you don't know the answer, but
yeah,
too many lockdowns, he's going to become more homogeneous, but is that better than still having a high, somehow developing high super spreading components?
Yeah, exactly. It's not clear. It's not clear to me, at least.
There's gonna be countries that do lockdowns and countries that don't.
Okay, yeah, I guess you're not modeling like that, yeah, but yeah.
No, exactly. So, but yeah, this is sort of very isolated, one factor thing. But yeah, okay. So this is just what we just talked about. This is the same stuff, but just that to visualize how that if you are in a situation where with some limited number of contacts, then you can really compensate for being on average not very good at spreading by being more homogeneous under the lockdown. So just to make more clear, I can say that if you have the ancestral variant or the good old COVID classic here, then you could, in principle, you could actually you could sort of like essentially, or maybe I should go down to the bottom plot where it's even more extreme with 10 contacts. Then you could actually halve the mean infectiousness and keep the same effective sort of average number of infections by just being homogeneous. So that's what you can see. You can sort of follow this contour, right? That you can just increase K, so that means decrease super spreading, while decreasing your mean biological infectiousness, and it actually doesn't matter for your ability to effectively spread in the population under the lockdown. This is just another way of putting the same thing. But then let's just imagine, just very quickly, that we actually let the variant just let it evolve freely, so to speak. And this is just a bit exaggerated, but let's imagine that you have some disease, and every time that it infects someone, there's a chance that it will mutate. And in this case, I just assume that it will mutate in its degree of overdispersion or the super spreading tendency with some probability. And I assume that's a geometric Brownian motion that's completely unbiased. So either you go a little bit up by some factor or you go down by some factor. So if you assume that, then any kind of sort of drift that happens or any systematic change in the super spreading tendency would be due to external pressures, because of course the mutation algorithm itself is unbiased. So let's say you do that with some probability you mutate once it is passed on, this disease. And then what you see, maybe not super surprising, is that in a non-lockdown society, this is without lockdowns, the first one, so you see a big epidemic wave, then the average degree of super spreading among the pathogens in the population doesn't really change, or at least very little. So that's what the purple thing is. But once you're under a lockdown, it increases super rapidly. But of course, the actual rate of increase is fictional here, right? Because I just assumed some mutation rate that is not realistic necessarily. But I just want to show the concept that really you have an enormous pressure for the K, so the super spreading tendency to increase. And that simply happens just because that, it's just selection, right? That the strains that show up that have a more super spreading tendency, they don't really make it. But then you could, if you think about it for a little while, you could make an objection. You could say, well, Bjarke, you're cheating a little bit, because in the top plot, you don't have a lockdown. So that means that the disease progresses much more rapidly, and that means that there are many fewer generations of for the pathogen in which to evolve, actually, right? Because you have each person now infects maybe 3 instead of each person infects a little bit more than 1, and that means that the infection chains will become much shorter, become broader but shorter, and that means that you can't get as far genetically from the origin as you otherwise could. So you could say that it's a bit of cheating to compare these two, but then I would counter with the argument that, okay, then I make a situation where there is not a lockdown, so I don't reduce the number of distinct contacts that people have, but I just reduce the overall infectiousness of the disease so that it matches, initially at least, so the initial growth rate matches the panel B. It's just a sanity check. So that then I remove that effect of having shorter infection chains and fewer generations. And then what I see is that the increase in K is still quite low, like in the first picture. So what that shows us, just to recap, is that it is really the lockdown that is pressuring the disease to grow more super spreading in this kind of model. It's not just the statistics of having fewer generations in which to evolve in a non-lockdown scenario and stuff like that. So it's in this simple model, the lockdown really does provide a pressure on the pathogen to evolve. But that's essentially the end of it all. I think for me, that's the last thing I wanted to show. And I think the main message for me, apart from all the super spready stuff, is really just that, these heterogeneities in epidemiological modeling are super rich. In a sense, there's so much that you can look at there, and that hasn't really been studied so much, because usually the state of the art in studying heterogeneity and these kind of things usually is to sort of age structure society or structure into multiple compartments. And that works for some things, but for something like super spreading, it doesn't work that well. And you can even think, why doesn't it work? Well, imagine the gamma distribution. So that had some K parameter that I said was 0.1 for COVID, but it's also a fact that if you sum gamma distributed variables, then you also sum the dispersion parameters. So once you are summing 1,000 different gamma distributed variables, then you are having a K value of 100 in this case, if you started with 0.1. So that means that it becomes completely deterministic, and you've washed out all the heterogeneity, even by just creating a compartment of 100 persons. So if, as we've just shown, if these inter-individual variations matter, then you can't actually capture them in compartmental models at all, even no matter how small you make the compartment almost. You have to come very close to the individual level. So I find there's a lot of interesting stuff there. And especially I think that, it becomes interesting if you couple it with an evolutionary dynamic. And this was just super simple stuff, but you could imagine something much more elaborate, sort of thinking about phylogenetic inference. How is it affected by super spreading and those kinds of things. I think there's a lot of fun stuff to be done there, essentially. And I think that's all I have to say.
Thanks. Yeah, I guess we have time for some questions. If anyone has, please raise your hand online or ask.
Yeah.
Yeah, go ahead.
Sorry. Yeah, so thank you. This is very interesting. Just coming back to my earlier question on this value from the PCR tests, in a sense, if you in a super spreading disease, if you could quash, if you could actually grab and sort of lock up the people with super spreading, then you would have an enormous disproportionate effect, right? If you would grab them and put them in a room.
Yes, absolutely. Yeah, that's what sometimes known as cutting the tail or chopping the tail. Or something.
Yeah, no, exactly.
Yeah,
and
that's.
But that would be
completely right about it.
Policy conclusion to this, right? Is as soon as somebody has a high viral load, you should immediately lock them up.
Well, that's the takeaway message that some people have that's what some people have suggested. I see where that's coming from, but in general, it can be quite hard. You can't really, you often cannot really identify these people before they do their deed. But actually, my main takeaway message is, I think there's two levels to it. Let's imagine first that you cannot identify super spreaders for some reason. I mean, it's very difficult at least. Let's imagine you can't. Then it's clear that lockdowns have a disproportionate effect in the super spread disease, whether or not you can identify. So that's my one takeaway message. And actually, the other one that I think is really great has to do with that I cannot take any credit for really, which was in a paper by Endo et al. I think, has to do with contact tracing actually. That the way that we do contact tracing in Denmark, for instance, is so-called forward contact tracing. So you find someone who's infected, and then you ask, oh, this person who's infected, could they have infected anyone else? And that's, if you have super spreading, that's a terrible way of doing it, because we know that in super spreading, most people don't become infectious. But on the other hand, let's imagine this diagram on the right hand. If I find someone who's infected, let's say this yellow guy here or here, for instance, and I ask who might have infected this person, well, then I end back at the hub, right, with an enormous probability. And then I could say, who might that person otherwise have infected? So that's sort of this backwards, then forwards thing. And they do that in Japan, for instance, and it works so much better. So that's also a takeaway message, I think, that you should really leverage the infection network structure, that it is so so characterized by hubs, that you shouldn't do forward contact tracing essentially.
What do they do in Denmark? Is it backwards or forward?
No, in Denmark they do forward, unfortunately. Yeah. So and it doesn't work that well. Yeah. So, but we have to actually talk to them about it a bit, and I think they are interested in doing the backwards type. But in Japan, they got this quite early. I think it was smarter to do it the other way around. Yeah, because then you move back to the hub, right? Then you find the guy who's really infecting a lot. Mm-hmm. So, but of course, you also write, Roland, about if you could chop the tail, then that would be really marvelous. But then you need to answer this kind of question that was also asked earlier, like, who are these super spreaders? It's difficult. It's difficult.
Well, but you mentioned that you get them. So if there's a high correlation between the cycle value in the PCR test.
That there is. So you could say that.
That's a pretty solid parameter then.
But if you test people a lot, then you can do it essentially. Yeah. Yeah. And then you of course need to do a bit of testing. But yeah, that's true.
Are there more questions? Or?
Yeah, I have a question. Well, so there were several kind of heterogeneities that you discussed. Of course, the most important was this dispersion parameter of the heterogeneity in the viral load within people, or the number of infected people that they did. And then on top of that, you have this network structure, which is also heterogeneous because you have clusters, etc. Some popular, well, at least I've also seen some talks of people that are discussing not only the network structure, but also higher order contacts. In that sense, could you get something similar, like if you have a suppose you have homogeneous transmission, but your social contact network has, let's say, nodes that are connected to, through higher order structures, let's say, instead of having just pairwise infection, you have some nodes that infect a large number of nodes in the same thing. Would you see similar results in that sense?
Well, it's a very good question, and it's actually a bit subtle, because that's also a viable model of some kind of super spreading, but it has another signature, because what you see is that if it's due to some people, as you're saying, having an enormous number of social connections, or being in large groups of contacts as opposed to pairwise and so on, then what you'll see is that the super spreaders will also be super receivers. So that means that the person who is highly likely to infect a lot of people is also highly likely to become infected. And that gives you a completely different epidemic signature when you sort of couple the two, because then what happens is that very early in the epidemic, all the super receivers will become infected, just
Yeah.
probabilistically, and then they will have an enormous reproductive number. So what you will see is that you will see a very rapidly rising epidemic that then suddenly dies out and doesn't come back, essentially, because you have depleted all the very highly connected people and so on. So that's a different thing, and something people have also thought about, and it's completely reasonable. But it's a different kind of signature you see then.
So people have ruled out that model. Is that what you mean?
Yeah, I think so. And of course, there's something to it, because of course there are different contact rates among people. That young people have more contacts than old people and so on. But I think it's fairly ruled out that it's what's driving the COVID super spreading thing. Yeah.
Right.
But it's a reasonable model of many other things. So yeah.
Another question would be like the mean. So is it possible to incorporate this heterogeneic spreading in some form of a mean field approximation, which goes beyond, let's say, standard homogeneous SIR as type approximations, but just including this picking
Yeah,
the rates from this gamma distribution.
I don't actually think so. I think this is one of those cases where actually the mean field approximation just breaks down. That I don't think there's any way of doing it.
But why not? I mean, why can't you just choose a varying rate in these things and then and then do some mean field on that? Why is that not allowed?
It's definitely allowed, but you're just not capturing the things that we're seeing here. That if you just assume that the infection rate in the entire population, for instance, is just time varying, then you don't get these effects of the sensitivity to network structure, and you definitely don't get what we see here is that much of it is also driven by local saturation. So that means that if you are highly infectious, then you will quickly infect the people around you, for instance, and then in the next few time steps where you are still highly infectious, you have run out of contact, essentially.
No, but I guess
you cannot have that kind of thing.
Yeah, but I guess you it's not like making it depending on time. It's more like making it depending on space, right? If you can introduce a space label and then have the the infection rate varying with space, couldn't that?
Well, you could do that, but of course, space in this case is also just another contact network, right? That if you have space, then let's say you're putting people on a lattice, for instance, then
exactly.
that's just a particular kind of network, right?
Yeah, right.
And then you could say,
go ahead. Sorry.
Then you're actually back to the agent-based model, I would say, because then you're saying, here's a guy who's highly infectious at this lattice point. Well, then you're doing agent-based modeling more or less.
Yeah, but I'm trying to do it more like a continuum approximation to it, right?
Yeah.
I don't know if it gives anything.
Yeah, that with space you could perhaps do it. But with the classical compartment models, you can definitely not do it.
No, that I understand. Yeah,
you can do there are mean field models on networks that take into account this heterogeneous structure in networks, right? So
yes, but this is not primarily driven by network structure, as we saw in the. I mean, of course, network structure plays a huge role, but for this,
putting the network is not a problem. The problem is putting the varying infection rate per node.
Yeah.
Right.
Exactly.
Is what he's saying, and then you
and the interaction between those. Sorry.
Yeah, we get it. Yeah, but.
Okay.
It smells like it should be possible to to enlarge the regular model to some sort of space-dependent kind of thing, and then I don't know. I mean, sounds like could be. But,
yeah, it's at least worth thinking about. But I think the space thing will very quickly end up being a little bit equivalent to this kind of thing. That then you're just using the position as an index instead of the whatever.
I'm not saying we're going to get anything new, but is a reformulation.
Yeah, but that's also interesting. That's
Yeah,
also
yeah,
interesting. At least it's sometimes nice to have some equations. I know some people tend to feel so. Yeah, that's true, but it's worth thinking about at least. But also the other thing is that it's amazing that quite a lot can be captured by just this really simple statistics, of course. But
yeah, right. Yeah, you showed you could basically get
this
the
stuff was just
result
from that. Yeah, exactly. So that is definitely true.
Yeah,
it's worth thinking about.
You could call it a day here, and
In principle, you could, yeah. In principle, you could, yeah. That's true. But I still thought it was fun to get sort of a bit of network structure on top of it. But you could say it's just the icing.
No, but I understand. Yeah, I understand.
Yeah. That is true. But I have this is of course a more philosophical debate, I guess, but I find it's always really nice that you can do things in two ways. That it's sort of I believe things more if you can sort of write down a really basic agent-based thing, and then you can also write down you know, some statistical or some equations or something that actually agree somewhat. Then I tend to believe stuff. So,
yeah, I guess you need both. Yeah.
At least it's like having experiments and theory, right? But then I'm just running an in silico experiment to some extent.
Right.
So yeah, at least that's a very positive spin on it. But yeah.
All right. Are there any more questions, or that's it?
I also realized I may have run a bit long, so that's.
Yeah, no, but you got a lot of questions, so it's not your fault. Yeah, it's not your fault.
No, it was great. It was super cool. So
Yeah,
thanks.
it was very nice.
Very nice.
All right.
Very clear. Yeah.
Yeah. Thanks a lot. Yeah.
But I guess I'll hang around for a little while if anything, if anyone wants to discuss something, or we talked about something maybe, Jay.
Yeah. I don't know. Yeah, I think. Yeah, I kind of. Yeah, made this the discussion.
Fair enough.
But we can talk a little bit more just about this idea maybe. But yeah, let's thank Bjarke again. I'm gonna switch off the record.